Hello everyone, we are from group 6 of the Nano SCL main grouping, and now we start with our introduction.
My name is Christina Tiong with metric number 2055669.
My name is Yeoh Yi Ying and my matrix number is 205228.
My name is Ooi Yi Lin and my metric number is 206904.
Hi, my name is Jun Ying and my metric number is 2066662.
Hi, my name is Ooi Zi Wei with matrix number 207281.
Okay, so now we start our discussion from group 1. I will be sharing screen for group 1.
Okay, yeah, so for group 1, I will be actually sharing on the first question, and Jessie will be sharing on the second question later on. So I'll be sharing on the question on how can nanotech and nanomaterials contribute to personalized medical. So firstly is due to the surface modifications property. This is true by inserting inert materials such as PEG to reduce the toxicity at the nanomedical medicine. Next, it also helps to reduce the side effects of the personalized medicines. And lastly, it also increase the specificities by adding biomolecules that is specific to the target cells or molecule. And secondly, due to the structure modifications property, it can improve the diagnostic by adding SP ions inside the nanomedicines by providing mechanical properties. Next, it also enhances the biocompatibility of personalized medicine by adding biocompatibility ions. And third is also due to its control release property, which is also known as CRS. It can enhance the efficacy while minimizing the side effect. It also delays the time taken for individual for the next treatment release. And lastly is it can act as a biosensor and diagnostic tools. Means for example, nano sensor can detect biomarkers of early stage of disease, allowing the early diagnosis. And for example, it's like nano sensor device for the brief acetone detections. So next I will be passing to Jessie.
Thank you, Ziwei. So now I'll be sharing about personalized medicine for patients who have prostate cancer, but it allergy to the docetaxel drug, which is a type of drug to treat the prostate cancer. So based on this is the simple structure of our design. So first of all, we use the drug named as cabazitaxel. So it is a type of drug to treat the prostate cancer, and this is the structure of the drug. And because the target patient is Asian, so we need to reduce the dose of this drug to prevent the side effect. And we also use a tyrosine kinase inhibitor, which is cabozantinib, and it is used to increase the efficacy of drugs towards Asian. And we coat this drug with the nanomaterial, which is PLGA. It is a biodegradable and biocompatible nanomaterial to load the cabazitaxel. And lastly, we use a receptor, which is PSMA ligand, to target specifically on the PSMA receptor on the prostate cancer. So this is all from our group. And do you guys have any question?
I have question group 1. May I know how does tyrosine kinase inhibitor increase efficacy of the drug?
Thank you, Ying. So for the tyrosine kinase inhibitor, so when we combine the tyrosine kinase inhibitor with other drugs, can result a synergistic effects of the drug, where the combined effect of both drug is greater than the sum of the individual effects of each drug. So this synergy may lead to the enhance of the anti-cancer activity.
All right, thank you.
Group 1, I have another question. So for personalized medicine, so is that important for them to have the magnetic properties?
Okay, thank you for the question. So, it is actually important to actually incorporate the biomagnetic properties into the nanomaterials for personalized medicines, because this magnetic property, it can enhance the versatility and also the applicability in the personalized medicines that allows it to be more precise and targeted towards certain cells, and it also able to improve the efficacy and the safety of the treatments and minimize the side effect.
Any other question? If no, I think we can proceed with second group.
So can you guys see my screen?
Yes.
Okay, so I'm Jun Ying, and me and Yi Lin are from group 2. So our topic is about smart farming. So smart farming is an advanced concept used in agriculture sector with the aim to increase the crop yields, improve food safety, reduce the rely on human resource, and finally to promote the sustainable agriculture. So in agriculture sector, you can use nanotech to control the release of fertilizer or drugs, and the drug or antibiotics can be incorporated with nanomaterials, so for the better efficiency. And based on the idea told by Dr. Jeff, our group have come up a nanomaterial design that incorporate the crab molting hormones, SCD steroids, into the nanostructured lipid carriers, NLC. So this is to solve the problem that the conventional molting induction method is harsh and low productivity. So the hormones will be encapsulated into the NLC, then mixed with the crab feed formulation, and then finally to produce them into the pellet form.
So let me continue. So actually NLC are a combinations of the solid and liquid lipids that are biodegradable and compatible. So it can encapsulate a variety of drugs, including hydrophobic and hydrophilic compound. The dual lipids matrix in NLC that you can see in the figure there, it allows for higher drug loading capacity and improve the encapsulation efficiency. The presence of the solid lipids enhance the stability of the NLC, and NLC also offer control and sustained drug release, providing a more predictable and prolonged release profile. So by loading the HCD steroid into NLC, we can save times and production costs, as the hormones can shorten the molting process of the crabs. So besides that, this method can also maintain a healthy wild crab populations and also allow sustainable farming. Besides that, the manual labor can also be reduced because NLC can slowly release the XCD steroid to control molting. That's all from our group. Is there any questions?
Hello, group 2. I have one question, which is, will the surfactants on the outer layer of the NLC poses a toxic effect to the crab or human?
All right. Thank you, Phua, for the question. So yes, of course, if you use chemical surfactant, I think it will possess toxic effect, especially when human consume the crab. But I think we can use biosurfactant for this purpose. And based on my limited knowledge about biosurfactant, it can be obtained from biological source. So it can be more suitable for in vivo application. However, I think we need further study regarding this because the surfactant is important to maintain the stability of the NLC and to ensure the efficiency of the hormone in the NLC. Yeah. Any other question?
I have another question. May I ask why use NLC instead of SLN?
Okay, the reason why we use NLC is because NLC has greater stability and the drug loading capacity, and also it has lower toxicity compared to SLN and offer a high control release profile that can reduce the side effects and enhance the therapeutic efficacy. Any other questions? If no, let's continue with the next group 3.
So I am from group 3. The activities that we get is related on the use of nanotech in cosmetic and beauty product. So the development in nanotech has led to the innovation and improvement in beauty and cosmetic product. This mind map shows some contributions of nanotech in those products such as sunscreen, lipstick, and nail polish. The first one is that the small size increase the efficiency of transport of the active ingredient for better absorption, and also the sustainable release of active ingredient by using nanomaterial reduce the frequencies of applying the products. It acts as the protective agents to enhance the stability of the molecules inside. Besides, the optical characteristic of nanomaterials also provide new color elements by adjusting the size of the nanoparticles. By using the appropriate nanomaterial, synergistic effect will also increase the functions of those ingredients packed in the nanomaterial. So for the question 2, our group also have developed a product which is lotion by using nanotechnology. This lotion is focusing on whitening of the skin. Therefore, we decide to use lacase as the active ingredient which can break down the melanin. In order to increase the stability of the lacase, solid lipid nanoparticles is used to cover the lacase. By the adding of solid lipid nanoparticle, lacase can cross the skin barrier and diffuse into the skin easily. Specific receptor that targeting the melanocyte was designed on the surface of the solid lipid nanoparticle to ensure evenly disperse of our product based on the severity of darkness caused by melanins. I think that's all for my group. Do you guys have any questions?
Thank you, Group 3, for your presentation. So I would like to ask, why would your group prefer to use SMP as the nanomaterials to pack the lacase?
Thank you for your questions. So the main reasons that we choose to use solid lipid nanoparticle as the nanomaterial is due to its efficiency in skin delivery, as it can cross the skin barrier to reach the cell that located below the skin easily. Besides, lacase is a hydrophobic enzyme. Therefore, it is suitable to pack them inside the solid lipid nanoparticle, which is in contact with the hydrophobic tier of the solid lipid nanoparticle to protect them from the aqueous environments. Yeah.
Thank you for the answer.
Is there any question? If not, I think we can proceed with the activity form. Thank you.
Okay, let me share my screen first. Okay. So, I'm from group 4. So just now I have heard your presentation. So it's great to learn about the various applications and of nanotechnology and its innovative contributions. However, every technological advancement comes with its own set of pros and cons. So in group 4, we have discussed on the potential issues associated with the use of nanomaterials at the aspect of medical, environmental, and social economy. Measures to each impact are also suggested. So this is the mind map for our group. So we have listed many points, so let me summarize for all of you. Let's take the example of titanium dioxide in sunscreen to discuss the potential medical concerns and environmental impacts. Given that titanium dioxide is in the nanoscale, have you ever considered the possibility of it penetrating into our skin? So the penetration of titanium dioxide into the skin might lead to the generation of reactive oxygen species, causing oxidative stress to the cells and potentially resulting in cellular damage. So this can cause inflammation and allergy reactions to our cells. In the case of spray products, inhalation of titanium dioxide may lead to lung inflammation. Additionally, when sunscreen is applied during water activities at the sea, titanium dioxide may enter the water, posing a risk of toxicity to aquatic ecosystem and eventually contributing to the bioaccumulation of titanium dioxide in the food chain. As for socio-economic impact, concerns arise about the potential monopoly in the nanoparticle market, which could widen the existing gap between the rich and the poor. To address these issues, a comprehensive regulatory framework is essential. So this framework should cover the safety standards for nanoparticle production, proper disposal of products containing nanoparticles, and also prevention of market dominance. So establish such measures at the administrative level is crucial to ensure the responsible and safe use of nanotechnology. So this is all from me. Anyone have any question?
I have a question. Apart from what you have shared, any other issues does the small size of the nanotechnology contribute to?
Okay, so due to the unique properties of nanotechnology, particularly its small size, our group has actually discussed, and we think that it might raise concerns about the potential for misuse in the context of biological weapons. The nanoscale dimensions make it challenging to detect, and its use could facilitate a rapid and widespread impact. So, moreover, the unpredictable delivery routes further add to the complexity of addressing and mitigating such threats. Yes.
Okay, thank you.
Any other question?
No.
If no, then I think this is the end for our presentation. Thank you.